This is a production of Cornell University.
First, I just wanted to thank Margaret for inviting me to this early career seminar. It's a huge honor to be an invited speaker as a graduate student, so I'm super excited to be able to tell you guys a little bit about my thesis work studying protein-protein interactions in the Arabidopsis circadian clock. So circadian rhythms, let's see here. Circadian rhythms are defined by these three primary criteria. They're endogenously generated, so they are encoded in the genome. And they produce 24-hour rhythms and are temperature compensated, which just means that these 24-hour rhythms aren't going to speed up when it gets hot outside, and they're not going to slow down when the temperature drops. And circadian clocks are present in all kingdoms of life and regulate various important phenotypes. So in humans, we think of the sleep-wake cycle. In plants, we often think of flowering time, and the clock is even in cyanobacteria to control the timing of nitrogen fixation. And in a big win for the plant biology field, the circadian rhythms were first identified in plants in 1729 by this French chronobiologist who noticed this interesting leaf movement behavior in one of his plants outside his house, where the leaves lowered and raised every day. And so he did a simple experiment where he brought his plant inside his house into his basement and put it under constant darkness. And under this condition, he noticed that this leaf movement behavior continued with a period of approximately 24 hours. And this was the first evidence that, that plants had a circadian clock. And so the way this works is that this plant is given an input cue from the environment, and in plants that's primarily day length and temperature cycles, which feed into the core oscillator, which I'll talk about in the next slide. And this oscillator coordinates a physiological output response, which here is leaf movement. So when you break the clock, these clock regulated outputs are desynchronized. So leaf movement might have a shorter period than 24 hours, it might have a longer period, or as shown here, it might desynchronize to the point of arrhythmicity, where you lose rhythms altogether. And the clock controls more than just leaf movement, of course. So cold tolerance, flowering time, even biotic stress response and pest resistance is clock regulated. So all of these phenotypes are actually have a time of day component associated with them. So this figure represents the core oscillator in Arabidopsis. And so it consists of these interlocking transcription translation feedback loops. And so I'm not going to walk you through the very complex signaling pathway pictured here, but what I want to demonstrate is that this complex signaling pathway operates to produce these very precisely timed phased patterns of gene expression over the course of the day. And it's this time of day specific gene expression that produces these time of day specific phenotypes. And one thing about the clock oscillator is that all of these components have been identified by Forward genetics. And so in the Nusnell lab, we take a proteomic approach to identify new circadian clock components and even connections between environmental input pathways and physiological output pathways. And the way we do this is using a method called affinity purification coupled with mass spectrometry. And so in this method, we're using Arabidopsis, where we've engineered it with a clock gene of interest driven by its endogenous promoter. And we add two affinity tags, a 6x histidine tag and a 3x flag tag. And you might hear me refer to this as the HFC tag for his flag C terminal. And we engineer this in a clock mutant background. So whatever gene we're interested in, we're engineering that into the clock mutant background. So our affinity tag version is the only one operating in that plant. We make protein extracts from that genetically modified plant, and we perform two affinity purifications. The first is purifying for that flag tag. And so our clock protein, along with any interacting protein, as represented by these colored triangles, and that can be a direct interaction or an indirect interactor, those proteins are co-purified. We perform a second affinity purification, isolating for that his tag to further to further purify specific interactors for our clock protein, while non-specific binding proteins will flow off. We've identified these interacting proteins by mass spectrometry, and then we perform a quality control step in which we're subtracting out any proteins that co-purify with our negative controls. The first of which is a GFP with our affinity tag, and we also perform a wild type no tag control, because those experiments are only going to pull down non-specific binding proteins that we are not interested in. So we've performed this method for about 13 of the core circadian clock components. And along this timeline, I'm showing the time of day that we collected tissue for each of these proteins, which corresponds to the time of day of peak protein expression. And for each of our affinity tag lines, we ensure that they meet two criteria. We first confirm that our HFC version of the tag of the protein is operating exactly like the native protein would. And so, for example, for Rev8, we have line 11-3, which rescues the long circadian mutant phenotype of the Rev8-1 null mutant back to wild type levels. Another thing that we check before we can use these lines for affinity purification is we do a 24-hour diurnal time course to look at protein expression over the course of the day to ensure that the circadian expression pattern is present. And also, this is how we identify the time of day that we're going to collect tissue for our experiment. So for Rev8, it has a maximal peak around 6 hours after dawn. So we've performed this process for all of our affinity tagged lines at this time. And I also wanted to acknowledge help from our collaborators, Dr. Norihito Nakamichi, who provided our PRR 5, 7, and 9 lines, and Dr. Andrew Miller and Dr. Uriel Urquiza Garcia provided the talk 1 line. And while I'd love to show you all of the very interesting things that we found in all of these lists, today I'm just going to be focusing on link 1, link 2, and Rev8, as those are the core of my thesis work. So link 1 and 2 are night light inducible and clock regulated 1 and 2. And they are transcriptional co-regulators with the activator transcription factor Reveille8. And together in a complex, these proteins activate the expression of evening circadian clock genes. And today I will mention TOC1 and PRR5 later in my talk. So those are the two that I want to highlight here. And a previous study demonstrated that the links perform this co-activation role by recruiting RNA polymerase II to Rev8 cis regulatory motifs in target gene promoters called the evening element. Okay, so I'm going to jump into a few what we found in these affinity purifications for these 3 clock proteins. So in this table, the values that you're seeing are exclusive unique spectra. So these are the spectra that were identified in the mass spec for these interacting proteins listed here. And this is, of course, a very abbreviated table. I'm just pulling out 4 of these cold response proteins that we noticed were very high up on our interactor list in terms of exclusive unique spectra. And so these are TCF1, which stands for tolerant to chilling / freezing 1, a family member of TCF1, which is a regulator of chromosome condensation family member, and then core 27 and core 28, which are cold regulated proteins. And so because we identified these cold response proteins so high up on our interactor lists for this transcriptional complex, we hypothesized that this link Rev8 transcriptional complex could act as a conduit between the clock and cold temperature input and or regulation of cold temperature as an output of the clock. And recently we got some encouraging evidence that we were on the right track. So this 2021 paper came out demonstrating that the Reveilles are positive regulators of the DREB1 transcription factor family. These are also known as the CBFs. And so they demonstrated that in particular, Reveille 4 and 8 are important for activation of DREB1A. And additionally, in figure D, you can see that the circadian expression pattern of DREB1A is severely dampened in these Rev mutants. And they further demonstrated that this has a physiological consequence in the plant. So when you knock out Rev4 and 8, the plants perform worse in freezing tolerance tests, as demonstrated in B and C. Okay, so when I had this list of proteins, I decided to focus on core 27 and core 28, primarily because when I looked in the literature, I saw that the cores were already associated with circadian rhythms previously. So in this 2016 paper, the authors demonstrated that the cores have a long period mutant phenotype when you knock them out. This is very similar to what we see in the link mutants, indicating that the cores and the links could be operating in the clock in a similar fashion. Additionally, very excitingly, this paper demonstrated that the cores are repressors of PRR5 and TOC1, those two evening express circadian clock genes that are activated by the link rev transcriptional complex. And specifically, the cores appear to be repressors of these genes at the end of the day towards dusk. So there's a time of day specific component there as well. And just a note that the link rev complex is activating towards more the midday morning. And then they also demonstrated that the core mutants exhibit enhanced freezing tolerance. So these overexpression lines here at the bottom perform worse than the wild type here, while the mutants perform a little better in these freezing assays. Okay, so you might have noticed from my interaction diagram that the cores appear to be link 1 specific interactors. But we were curious if this was truly a link 1 specific interaction, or if we were simply not seeing the cores in the affinity purification experiments from Rev 8 and link 2 because of the time of day that we collected tissue for those experiments. So again, we collected tissue based on the time of day of peak protein expression for our bait proteins. And so for link 1, that was 9 hours after dawn, while Rev 8 and link 2 were collected 5 hours after dawn. And so I'm going to represent this graphically here. So I've mapped onto this figure the protein expression patterns based on these 24-hour Western blots that I showed you earlier, just with densitometry analysis for our circadian clock proteins. And if I overlay onto here the mRNA expression patterns for core 27 and core 28 from a public database, you can see that the cores have a circadian expression pattern and they peak towards dusk, about 12 hours after dawn. And if I map onto here the time of day that we collected tissue for affinity purification, you can see that at ZT5 or 5 hours after dawn, the cores are barely expressed, while 9 hours after dawn, they're much more highly expressed coming up in the day. And so it's entirely possible that the time of day that we collected tissue is influencing our data. And so what we did was swap the time of day that these proteins were collected. Okay, so again, in our original experiment, we did not see any spectra popping up in our link 2 and Rev 8 affinity purifications. And then when we swapped them, we saw exactly the opposite. So when Rev 8 and link 2 are collected near the end of the day, core 27 and 28 now appear in their affinity purifications, while link 1 lost its interaction with the cores, indicating that there's a time of day specific component to this interaction. And this is interesting in the circadian world because this kind of narrows down the time of day that the cores could be influencing clock activity, specifically towards dusk at the end of the day. Okay, so the next question we asked was whether the links have a function in using temperature as an entrainment cue or a synchronization cue to the circadian clock. And to answer this question, we're using a luciferase reporter assay. And so that's pictured here, where we have a clock gene promoter driving the expression of luciferase. And this is just an easy way to monitor clock activity. So when we're imaging these plants, we first grow them in constant light. So this is a condition in which the plant has no entrainment cue. It's relying solely on its circadian clock to maintain rhythms. We then transfer it into a temperature cycle condition where we're providing 12 hours of 22 degrees Celsius and 12 hours at 18 degrees Celsius, in which a wild type plant would be able to use this cycling pattern to entrain circadian rhythms. And our hypothesis is that the link mutants would not be able to use temperature cycles as an entrainment cue. Okay, so again, in this first part of the experiment, we're in constant light. And as I mentioned in the introduction, link mutants have a long period phenotype, which you can see nicely here as as the rhythms get further and further away from the wild type as represented in black. And to kind of help you look at this data, I've traced on with this dotted line the peak expression of wild type luciferase reporter. And so if you look, for example, at the link 2-4 null mutant, you can see that the peak of link 2 gets further and further away from wild type because of its long period phenotype. When we supply these plants with a temperature entrainment cue, you can see that it takes several days for these plants to use this information to entrain their circadian clock. And so I'm going to first walk through link 2 in pink. And so again, this peak is not overlapping with the wild type peak, indicating that it's not re-entraining to wild type rhythms. By the end of the experiment, you can finally see that link 2 has realigned with wild type, but it took several days for it to get to that point. In contrast, link 1 never realigns its peak expression with that of wild type, indicating that these mutants are impaired in their ability to use temperature cycles as an entrainment cue. And I think this becomes very apparent when we contrast this to where we're adding a photo entrainment cue after providing constant light. So again, our link mutants have this long period phenotype in constant light. And then when we provide just one night of darkness followed by dawn lights on the next day, you can see that all the link mutants immediately re-entrain their peak expression pattern with that of wild type throughout the end of the experiment, indicating that these plants aren't impaired in general in their ability to use entrainment cues to synchronize the clock, but it's specifically a temperature defect that they're unable to use. Okay, so based on the data that we've gathered so far, we've put together the following model, where in the late morning, about 5 hours after dawn, link 1 and 2 are interacting with Rev8 to fulfill their canonical role as co-activators of evening expressed Rev8 target genes, like PRR5. In the evening, we believe that core 27 and 28 are interacting with this transcriptional complex to prevent activation of these target genes, possibly by sequestering this complex or another mechanism. So the next thing on the horizon is characterizing the molecular mechanism of this interaction. So what is the functional consequence of the core link Rev8 interaction? And again, we think that binding of the cores to the link Rev complex could reduce the binding affinity to target gene promoters. So two questions that we're going to ask are, can core 27 and 28 bind Rev8 target genes without Rev8? So again, the cores do not have a DNA binding domain. So we're unsure how these proteins are associating with these promoters. And we think that Rev8 is likely the intermediate there. We also want to know, does Rev8 DNA binding activity increase in the absence of the cores? If the cores are engaging in this sequestering activity, I think we can answer it. We can answer this question. Both of these we're planning to use chromatin immunoprecipitation with qPCR to answer. Okay, so in summary, we've discovered novel interacting partners for 13 of these core circadian clock oscillator proteins using our proteomic approach. We've also identified a time of day dependent interaction between the link 1 to Rev8 complex with core 27 and 28 at the end of the day specifically. And third, we've identified a role for the links in temperature entrainment of the clock. And with that, I would like to thank everyone in the NewsNow lab at the Danforth Plant Science Center, our collaborators who've graciously provided reagents and lines for us, and my thesis committee, and I'd be happy to take any questions.
So you can either unmute yourself, or if you have a question that you want to type into the chat, I will read it out. Thank you. That was really great. And also provides another thing to be concerned about when you're trying to look for molecular interactions. It's nice that the protein interactions are also influenced by time of day.
Yeah, that's the circadian like researcher favorite question at a seminar is like, what time did you do your experiment? So, you know, it does matter.
Yeah, so I am less familiar with the temperature entrainment. I was wondering, are plants that are temperature entrained also showing the circummutations, the leaf flapping?
Yeah. So even without any light dark cycles, temperature alone can entrain circadian rhythms in leaf movement and all the other circadian outputs.
Cool.
Yeah.
Great. Yeah, and if you have any follow-up questions, you can contact Maria, or if you have a meeting already scheduled. Yeah, that was fantastic. Thank you.
Thank you guys for listening to my talk.
Cool. And now I'll introduce Jim Fouracre, who will tell us about phase change and development. So the longer term temporal regulation in plant development.
Yeah, thanks, Margaret. So there you go. Margaret only invites speakers who like moss and running. I think that's a clear theme. So yeah, as Margaret mentioned, I like to shift from a kind of a daily proteomics approach into a more developmental genetic approach with this talk. And so what I'm really trying to understand is our developmental clocks. When I talk about developmental clocks, I'm talking about the timing of developmental transitions. So flowering plants go through a series of different stereotypical transitions as they progress through their life cycles, from embryogenesis to seed maturation, and following germination, from juvenile to adult periods of vegetative growth, before the onset of flowering, the production of gametes, and the starting of a life cycle again. The developmental transition I'm most interested in, the Potig lab's most interested in, is this switch between a juvenile and an adult stage of vegetative growth, known as vegetative phase change. And what we're really trying to understand is, what are the factors that control the timing of this transition? So in Arabidopsis, well, depending on the species, vegetative phase change is associated with a wide variety of traits. In Arabidopsis, we've got a nice set of diagnostic features that distinguish between juvenile and adult phases of development. So juvenile leaves, for example, smaller, they're rounder, they have a flatter blade angle, and they also lack serrations and trichomes on the abaxial surface of the leaf here. Whereas adult leaves, they're bigger, they're longer, they have a narrower blade angle, and they produce serrations and trichomes both on the adaxial and the abaxial surface of the leaf. So it's a really kind of nice system to study phase change with these different character states. So in Arabidopsis and all other surveyed plants, vegetative phase change is triggered by the activity of SPL genes. And SPL genes are characterized by the presence of this squamosa promoter binding protein box, a bit of a mouthful. And a subset of SPL genes are also targeted by two microRNAs, so microRNA 156 and microRNA 157. And the binding site for these microRNAs can be in the coding sequence of a transcript or in the 3' UTR. And 156 is a more effective repressor, has more affinity for the target site than 157, but 157 does a pretty good job as well. So as these microRNAs repress SPL genes, and SPL genes trigger a switch to adult growth, we really think of these microRNAs as the master regulators of vegetative phase change. And there are 8 156 loci and 4 157 loci in Arabidopsis, and a subset of these contribute the most regulation, and 2 in particular, 156A and 156C, are the kind of crucial regulators. And you can see that in a 156AC double loss of function mutant, leaves start to produce abaxial trichomes, shown here in black, a much earlier leaf than in wild type plants. So these are really crucial regulators, 156A and 156C. And during development, the expression of these genes and the accumulation of the mature microRNAs decreases. So in this case, the mature microRNA decreases most drastically from 1 to 2 weeks of development, and then this decline slows a bit afterwards. And this accumulation pattern of the mature microRNA is really largely explained by the expression of the two primary transcripts, A and C. And they have similar but not quite identical expression patterns in that C declines a bit more slowly than A. And it's this temporal decline in 156 that really kind of underpins our understanding of how phase change works, in that the temporal decline in 156 expression leads to a concomitant increase in the accumulation of targets in the SPL gene family, and it's this increase in SPL products that triggers the switch from juvenile to adult growth. And this precedes the subsequent switch to reproductive growth following inductive conditions. So it's this temporal decline in 156 is key, but the factors that regulate this developmental decrease in expression of a master regulator are really unknown. And it's a key question in the field is what controls the temporal decline in 156 expression? So recently, work in the Potig lab and others has shown that epigenetic silencing of 156 is really crucial to its expression pattern. So if we look at the balance between an active and a repressive histone mark, this active histone mark H3K27 acetylation decreases over time. This is at the 156C locus from 1 to 2 to 3 to 5 weeks. So as 156 expression decreases, it's associated with less of this active histone mark. And likewise, you have an inverse pattern for this repressive histone mark, H3K27 trimethylation, which increases gradually from 1 to 2 to 3 to 5 weeks. So it looks like there's this balance of active and repressive histone marks is critical in the regulation of 156C. And when it comes to epigenetic regulation, we know that there are a couple of key regulators that coordinate this. And in particular, the polycomb repressive complexes 1 and 2. And this cartoon just shows you the kind of critical components of PRC1 and PRC2. And PRC1 tends to act through deposition of another repressive histone mark, H2A ubiquitination. And it can also promote the activity of PRC2, which leads to H3K27 trimethylation. So PRC1 and PRC2 are really critical factors in epigenetic silencing. And it looks like they also function in phase change as well. So loss of the PRC2 component curly leaf, which encodes a methyltransferase. Loss of this gene leads to a significant decrease in the deposition of H3K27 trimethylation, and an associated increase in the expression of 156. And likewise, loss of the PRC1 components, AT-BMI1AB, these two redundant ubiquitinases, leads to a significant decrease in the deposition of H2AUB and also H3K27 trimethylation. So you've got PRC1 and PRC2 appear critical to the silencing of 156. So to kind of summarize what we know about the temporal regulation of 156, we know its expression decreases over time, and this is critical to the regulation of the timing of phase change. We know that the expression of 156A and C, these two critical loci, is associated with an active histone mark, and the repression is associated with two additional histone marks, and that PRC1 and PRC2 are critical in the repression of 156. So this is the kind of our understanding of how 156 is regulated at the moment. But what we don't know is what is the temporal signal that leads to the gradual repression of 156. So we know that epigenetic silencing is important. We know it's the coordination of two different, the antagonistic interaction of different histone marks, but we don't know what the temporal signal is that triggers the replacement of an active histone mark by a repressive one. And that's really what I've been trying to investigate the last couple of years and what this project is all about. So a couple of good candidates for components that might supply this temporal signal are VAL genes. So VAL genes are B3 domain transcription factors, and there's 3 VAL genes in Arabidopsis, of which two are most important. And Val1 and Val2 were originally identified in their role in a kind of preceding developmental transition in how they repress embryogenesis. So loss of Val1 and Val2 leads to the continuous production of embryogenic calli at both the shoot and root apical meristems. So if you lose Val function, you don't repress embryogenesis. Our kind of best understanding now of how Val genes function is via a subsequent developmental transition at a flowering. And we know that Val genes bind to their targets, these RY sites at the FLC locus, floral repressor. And when Val genes bind to RY sites, they promote the activity here predominantly of PRC2, which leads to the deposition of H3K27 trimethylation in FLC, the repression of FLC, and the transition to flowering. So they're crucial regulators of different developmental transitions throughout the life cycles of plants. And then we've got a bit of an indication that they might function phase change in that loss of Val1 and Val2 also lead to a reduction in H3K27 at 156A and C. So we know Val genes interact with PRC1 and PRC2. They're involved in developmental timing or developmental transitions throughout the life cycle of a plant, and they have somewhat of a role here at A and C in terms of their regulation. So it looks like potentially they were pretty good candidates for maybe supplying this temporal signal into the regulation of phase change. And while that work was going on to try and understand how Val genes are functioning at other transitions, I was carrying out an EMS mutagenesis screen to try and identify some novel regulators of phase change. And I found this lovely, cute little juvenilized plant. It's got lots of nice, small, round leaves. It's got a significant delay in the timing of phase change. And mapping this mutation revealed it to be, unsurprisingly, in Val1. And this mutation leads to the conversion of a really very conserved arginine residue right in the B3 domain of the DNA binding domain. And so it's a very crucial mutation in an important part of the protein. And we've called it semi-dominant because even in the heterozygous form, we see a significant delay in the timing of phase change. So it was quite a nice mutation to find. So with all this in mind, what I want to do is what I have done is systematically tested whether Val genes supply this temporal signal, whether they're the fundamental core temporal regulator of vegetative phase change. So to do this, the first thing I wanted to test was whether Val genes have a temporal effect on vegetative development, on the phenotype of plants. So to do this, I combined my nice new Val1 mutation and another null tDNA of Val1 with several different Val2 tDNA insertions to get over a problem of redundancy between Val1 and Val2. And you can see here that perhaps that these two different Val1 single mutants have an effect on phase change. They are significantly delayed in the terms of the timing of trichome production, but it's relatively subtle. Val2 single mutants, take my word for it, have no effect on phase change. When you combine Val1 and Val2 to make doubles, in all three cases, you see some really nice delayed phase change phenotypes. These are really juvenilized plants, particularly in the case of this Val1-2, Val2-1. All the leaves this guy is producing, small and round, they look very similar to the first two leaves of wild-type plants. So it's suggesting that Val genes really are critical regulators of vegetative phase change. And to try and get a handle on whether they've got a temporal effect, I quantified the length-width ratio of leaf blades in these various different mutant lines. So in wild-type plants, in silver here, you see a gradual increase in the ratio between the length and the width of the blade. So as more adult leaves get longer. In Val1 single mutants, you see somewhat of a suppression of this because they're juvenilized, their leaves appear shorter and rounder for longer, and an even bigger effect in Val1, Val2 doubles. And it's at a later leaf stage that you really see these effects manifest themselves. So it looks like they are at least temporarily regulating the phenotype of phase change. So the next kind of obvious thing I wanted to test was whether Val genes are regulating the temporal expression pattern of 156. So I quantified RNA levels in these different mutant plants. And in wild type, you can see here again in silver, this very characteristic temporal decline from 1 to 2 to 3 weeks of both 156 and 157. And for the val1 single and also the val1-5, val2-3 double, you see a general increase in the amount of these microRNAs, particularly for the double mutant here. But surprisingly to us, the temporal pattern is remarkably consistent. So even though we've really strongly eliminated val activity, you see an elevation of 156 and 157 levels, you still see this very robust temporal pattern, which was, I'll tell you, a little disappointing, but nonetheless, that's what it was. So the next thing I wanted to test was, okay, well, val1, val2 don't appear to, or they're not necessary for the temporal decline in 156, but do they affect the temporal patterns of histone deposition that I've talked about? So the first thing I tested was effectively the activity of PRC2 via H3K27 trimethylation, and in this case at the 156C locus. So this is a schematic of 156C, and you can see that dotted along the 156C locus are 5 RY sites, which are the DNA binding sites for val genes. And in terms of H3K27 deposition in wild type, again in silver, you see this very nice characteristic temporal increase in the deposition of its histone mark. And in the val1, val2 double, you do see a nice attenuation actually of this temporal increase. So it does appear like val genes are important for the temporal pattern of temporal deposition of H3K27. So I also wanted to check how PRC1 activity is regulated. And to do this, I looked at the H2A ubiquitin mark. And unlike H3K27, you don't see a temporal increase in this mark. It's kind of flat lines across development. Very interestingly, in the val1, val2 double loss of function line, you see a significant difference throughout development. And this is kind of really important in terms of how we understand epigenetic silencing in plants, in that the kind of canonical model of PRC activity from animals is that PRC2 activity, so H3K27, precedes the activity of PRC1. Whereas what we see here, and this is a kind of emerging model in plants, is that really PRC1 activity precedes that of PRC2. And that's kind of mechanistically shown here at this specific locus, in that you see a significant difference at 1 week for PRC1 activity, but that doesn't manifest itself until 3 weeks for PRC2. So that kind of nicely supports other findings in the field of epigenetic regulation. So the next thing I want to test in terms of whether val1 is temporally regulating 156 is, well, does val1 actually have a temporal binding pattern to 156 loci? So to do this, I used an HA tagged version of the val1 protein and looked for a chip qPCR analysis of val1 binding over time. So this graph shows you effectively the affinity of val1 for the 156C locus. In white is the wild type negative control, and in gray, the temporal series from 1 to 2 to 3 weeks of val1 binding. And what this shows is that firstly, val1 has a different affinity for different regions at the 156C locus, and it binds particularly strongly around this RY site and this area of the gene. But there is no temporal pattern of val1 binding. Again, slightly surprisingly, it's kind of pretty flat affinity during vegetative development, which is again not what we were expecting to find. To further get a handle on how val genes interact with the 156C locus, I went about interfering with the binding site, the RY binding sites. So I took a 5 kilobase fragment of the 156C locus and removed each of the 5 RY sites individually, and also all 5 RY sites in combination. And I took these different fragments and transformed them into a 156AC, 157AC quadruple mutant background. So this quadruple mutant background has all pretty much 156 and 157 activity eliminated. So it's a really nice background to do these experiments because it's very sensitive to any perturbations to 156 levels. So you can see that in a this quadruple mutant background, these plants produce trichomes very early on about leaf 3. In the empty vector control, there's no effect. In a nice positive control here, we have is the triple mutant, where you've got a functional 156C. So in here, you've got the endogenous 156C locus is intact. And you can tell that having this endogenous 156C locus delays development to about trichome production to about leaf 5. So when I take a 156C fragment with all RY sites intact and transform that, you see the same behavior effectively as the triple mutant. So that's a nice endorsement to show that, okay, the wild type 156C fragment I'm taking behaves like the endogenous locus. Deletion of all 5 RY sites individually significantly delays the timing of abaxial trichome production. And when all 5 RY sites are deleted in combination, you see an even greater delay in the timing of abaxial trichome production, showing that all 5 RY sites are functional, and that it's VAL1, even though we don't see a temporal perhaps pattern for VAL1 regulation, VAL1 is certainly affecting 156 activity. Again, to investigate this kind of temporal relationship between VAL genes and 156 levels, I quantified RNA levels in these different genetic backgrounds. So in the triple mutant, as you expect, you see this temporal decline in 156 levels because you still have this endogenous 156C. For the wild type version of 156C in the quadruple mutant background, you see this temporal decline. And there's quite a lot of variation here because these are different independent homozygous insertions. But when you remove a single RY site, perhaps the most important RY site, and all 5 RY sites together, you see a bit of a bump in terms of 156 levels. But again, you see this really consistent temporal decline in 156 levels. So this supports the earlier results from the loss of function studies in that VAL genes aren't, or VAL activity isn't necessary for the temporal decline in 156 activity. So I also wanted to make sure that these results, what I see here is because of an interaction between loss of RY sites and VAL1 itself. So I introgressed a loss of VAL1 mutation into these different mutant lines to test for an interaction between the 156 locus and VAL1 itself. So as you'd expect in a triple mutant, and where we have the wild type version of the 156C locus, loss of VAL1 leads to a significant delay in the timing of trichome productions because you still have all these RY sites intact. Surprisingly, when I also introgressed VAL1 into lines that don't have RY sites, so you think there's nowhere for VAL1 to bind, so you shouldn't see an effect of loss of VAL1 function, you still see a significant delay in the timing of abaxial trichome production. So even when there's nowhere for VAL1 to bind, losing VAL1 activity still delays phase change. And so the most obvious interpretation of this data for us, I think, is that as well as functioning through 156, VAL genes are regulating phase change via 156 independent mechanisms. And so to test this, and this is the final piece of this puzzle, I combine the VAL1 loss of function mutation with the quadruple mutant, where you haven't got any 156 and 157. And the kind of gross morphology of early development shows that the quadruple mutant phenotype is largely epistatic. If you look at the development, certainly of leaf 1 and 2 and the rate of leaf initiation, it looks largely like the quadruple mutant plant itself, suggesting that the activity of VAL1 is largely mediated by 156. But when you look at the developmental time series of leaf development, we see that actually, there is an independent role for VAL, a 156 independent role for VAL activity. So in this case, I'm looking at the angle of the blade base. So more adult leaves of wild type plants have a smaller blade base angle because they get narrower. The VAL1 mutation by itself elevates this. It's got a nice flatter base angle. In the quadruple mutant, in is this cyan, teal, whatever color this is, it's got a much narrower blade angle. But when you introduce the VAL1 mutation, you do see an effect even when there isn't any 156 activity. So this is showing really confirms that VAL genes are regulating phase change via the 156 pathway and also independent of 156. And to get an idea of what might be going on here, I introduced a 156 resistant version of SPL9, which is our favorite SPL9 gene, has the biggest effect on phase change, into VAL1 backgrounds. So any effect on this transgenic reporter is indicative of a regulatory interaction that has nothing to do with 156, because 156 can't regulate this modified transgene. So in wild type plants, you see a very strong expression of this resistant reporter throughout development. But in a VAL1 background, 2 different alleles here, you see a large suppression of the reporter, both in terms of the GUS staining and also the actual kind of delayed leaf initiation phenotype here as well. So this suggests that the 156 independent role of VAL genes is at least in part mediated by a transcriptional regulation of SPL genes themselves, independent of 156. So just to kind of conclude, so it seems like VAL genes really function largely as general rather than temporal regulators of 156. And so we can explain our phenotypes, I think, if you're looking at if phase change occurs at a certain ratio of active to repressive histone marks at the 156 locus, and it's this balance here. In wild type plants, this is the kind of ratio of replacement. And in VAL mutant plants, you still see the same temporal pattern of epigenetic silencing, but it's just shifted a little bit in terms of time. Secondly, that the temporal expression pattern of 156A and C is remarkably robust. So you get rid of all VAL activity, you get rid of VAL1 binding sites, you get what I haven't shown you here, you get rid of PRC2 components in a VAL1 background. You still see this remarkable temporal decline in 156 expression. So it looks like plants have evolved a really robust mechanism for buffering 156 expression. So it appears very important to plants that 156A and C have this temporal pattern of expression. Thirdly, that PRC1 precedes PRC2, and like I mentioned, this is important in terms of our understanding of how epigenetic silencing works in plants and how it makes it different to animals. And lastly, that VAL genes regulate phase change via 156 dependent and independent mechanisms. And this is kind of consistent. So VAL genes, they function at multiple nodes of the phase change regulatory pathway. And this is also the case in embryogenesis. They act at different levels, and in flowering time, they act at different levels of the flowering time pathway as well. So it looks like VAL genes, although they aren't necessary for the temporal regulation of 156, have been co-opted into a variety of different genetic networks in the control of the timing of developmental transitions throughout plant life cycles. They're really central kind of hubs in timing mechanisms. And with that, I'd just like to say thanks. This is a very outdated picture of the Potig lab, and thanks in particular to Scott and to Victoria here, who was a technician who was helping me with this project, and also to lovely Simone in Albert Einstein, who was helping me with some proteomics on this study that I haven't had time to talk about today. And if you have any questions, I'll be happy to take them.
Thank you, Jim. That was valid. Got it, fun. That was great. I think Mike has lots of questions. He put them in the chat, but I see he's unmuted himself, so I'll just let him ask directly.
Mm-hmm. Yeah, I guess I'm wondering whether or not the VAL1-2 doubles really have a more severe phenotype than VAL1 alone. So I saw there's variation in those allele combinations of VAL1-2 double alleles, and it seems like even some that had 2 exon insertions in either gene, like this one on the far right bottom, was not as severe as the one in the middle, VAL1-2 versus VAL1-1. So what's this variation?
Yeah, so that's a good question. So this is also why we think that this VAL1 allele is semi-dominant. So if you compare, so the VAL1-2 is a null tDNA, but the VAL1-5 is EMS mutation, is just a single base change, but has, as single mutants, has a much more severe phenotype than the tDNA null. But when you make double mutants with VAL2, you see a much stronger phenotype when you have the VAL1-2 null tDNA. So really, it's these two look more severe than this one. It's these two blue lines are Val1-2s, and this blue line is a Val1-5. So what we think is going on in the Val1-5 is that it's a mutation in the B3 binding domain. It's affecting how Val1 is binding to its RY sites. And as well as doing that, it's interfering with how Val2 is binding to RY sites. So when it's a single mutant, it's got a stronger effect. But when you eliminate Val2 activity, actually it's not as strong because it's not interfering with Val2 because it's not there. So it's really the two null tDNAs that have a bigger effect in the double form, if that makes sense.
That makes sense. And then the other question was this drop in these repressive histone markers. Do you see that also in Val1 single mutants? I may have missed that.
In the chromatin state, sorry.
Yeah.
Yeah, no, I haven't looked at the single mutants just because, yeah, I wanted to check. I wanted to actually find something. So, yeah, that's something.
Nice talk. You should submit this to FACID.
Yeah, you already have a bit of a phase change heavy plenary speaker, I think.
Jim, I also had a question. Oh. Yeah, I was curious what photo period you were doing these experiments under, and if you think that changing to like a short day, long day would influence the temporal patterns of the mRNA and RNAs.
Yeah, for sure. That's a good question. So we do certainly for the kind of, whenever we're testing from 1 week to 2 weeks to 3 weeks, we have to do it in short day conditions, predominantly because Arabidopsis flowers so early. So, you know, if Arabidopsis has transitioned to an inflorescence meristem about leaf, you know, day 11, day 12, so very early. So we want to suppress flowering in our developmental time series to eliminate that from kind of being a mitigating factor. So we always grow these in short days because in short days at 1 week and 2 weeks and 3 weeks, these plants remain strictly vegetative. So that's very important to us. But also, yeah, day length has an effect on 156 expression itself. And you see both in terms of light quantity and light quality, longer and more light represses 156. And so it appears that maybe this is, so there's some physiological work going on in the lab actually by a PhD student who's just finished up, and she's really been investigating how phase change is an adaptive response to light conditions and how younger plants are better at using different conditions with less light. So we think that's kind of related to that for sure. So
okay, cool.
Thanks.
Cool, thanks.
Nice. Yeah, getting two temporal people in the room together make things more complex. So I was wondering, miR-156 is one of the most conserved microRNA families in land plants. Are vals also super conserved, and how broadly present are these RY sites? Like, are they? Does it seem like vals are targeting, I don't know, 50 genes, or are they targeting thousands of genes and kind of broadly regulating the genome?
Yeah, so the ChIP-seq analysis came out last year, I think it was, looking at Val1 binding across the genome. Yeah, and it comes out with a lot of targets. So there are RY sites, there are only 6 base pairs sequencing, so you're going to get them pretty heavily distributed throughout the genome. In terms of but there's a very specific interaction between RY sites and B3 domain transcription factors. So Val genes are part of a much bigger family, but it's only really Val genes and their sister clade, FUS3, ABI3, and LEC2, which are regulated with embryogenesis, that actually bind RY sites. So it's a very unique interaction between a relatively small set of transcription factors and RY sites. But I think, yeah, this explains why Val1s are so have such important roles throughout plant development at various different stages. Is that, you know, it's a relatively simple genetic interaction, so it's kind of been easy, I think, to co-opt them into regulating embryogenesis, phase change, flowering time, probably other developmental transitions as well. I can't tell you how conserved Val genes. I mean, all flowering plants have got them. Outside of flowering plants, I'll wait until I start working on moss, and I can tell you. But in terms, yeah, I mean, the evolutionary role of 156 is something that's what I'll be doing in Bristol, and that's really it's an exciting kind of avenue. What's 156 doing in plants that don't make leaves?
Mm-hmm. Leaf-like organs, but yeah. Great. Are there any other questions? Well, thank you to both our speakers. Please take a moment to maybe do a clap emoji or turn on your video, and thank Maria and Jim for excellent seminars today. Yeah, it was great to hear about your work and catch up with both of you.
Thanks, Margaret. Thanks for having us. That's some great conversation.
Yeah, thanks.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.